Hi everyone, my name is Bian Li. I'm a postdoc from the Department of Biological Sciences at Vanderbilt University. The title of my talk is Predicting Changes in Protein Thermodynamic Stability Upon Point Mutation with Deep 3D Convolutional Neural Networks. The thermodynamic stability of a protein, often quantified by the Gibbs free energy of folding, is a fundamental physical property of proteins. It is determined by the intricate biophysical interactions between residues within the protein and the interaction of the protein with its surrounding environment. When an amino acid mutation occurs in a protein A that results in a mutant protein B, very likely the native interactions will be disrupted. Depending on how much the resulting mutant residue can compensate for the native interaction, the stability of the protein can increase or decrease, and this change in stability is often called the delta-delta G. I would also like to point out that because delta-delta G is a thermodynamic state function, meaning that the delta-delta G between A and B has nothing to do with how you go from A to B, but only depend on what A and B actually are. So it must hold that the delta-delta G from A to B, or the direct mutation, and that from B to A, or the reverse mutation, be equal in magnitude and differ only in the sign. And this is the anti-symmetry property of delta-delta G. We will come back to this anti-symmetry property shortly. As you might have already realized, delta-delta G is a very important quantity with applications in diverse fields such as protein design, drug discovery, interpretation of genetic variants, even in protein structure prediction and the investigation of protein folding pathways. As useful as it is, however, experimentally measuring the delta-delta Gs of all possible mutations in all proteins is simply impractical. So over the years, many computational tools have been developed with the functionality to predict delta-delta Gs. Some methods calculate delta-delta Gs by explicitly modeling the energetics of protein structural conformation, while a larger group of methods are based on machine learning where either only structure or sequence features are used as input, or both kinds of features are combined as input. And with many methods available, people have also developed meta-predictors that combine predictions from various methods. Here, we present a novel method called ThermoNet for delta-delta G prediction based on deep 3D convolutional neural networks. We believe that a limited set of manually engineered features might not be able to fully capture important structural information for delta-delta G prediction, and that unbalanced training sets may cause bias toward predicting destabilization because existing datasets are dominated by destabilizing mutations. In our approach, we use deep 3D convolutional neural networks to learn features directly from protein structure with minimal manual feature engineering, and we create a dataset balanced for both direct and reverse mutations to train our method. For the 3D convolutional neural network to work with protein structures, we treat protein structures as if they were multi-channel 3D images. Here's what I mean by that. Given a protein structure and a mutation site, we divide the space around the mutation site into a 16 angstrom grid of 3D voxels at the resolution of 1 angstrom. We then assign a saturation level to each voxel to reflect its biophysical nature based on the biophysical properties of its neighboring atoms. In this work, we consider 7 such biophysical properties, including hydrophobicity, aromaticity, and some others. According to this analogy, a protein structure can be considered as a 3D image with 7 property channels. And for this given mutation, we also generate a structural model for the mutant and parameterize the same grid of 3D voxels in the mutant structure. By doing that, we obtain 2 tensors, one from the wild-type structure and one from the mutant structure, that we can then stack together and use as input to a 3D convolutional neural network that consists of 3 hidden convolutional layers and 1 hidden fully connected layer. In the beginning, I mentioned that delta delta G has the anti-symmetry property. This means that ideally a delta delta G predictor should meet the following 2 necessary criteria. One is that the prediction bias delta, here quantified by taking the sum of the prediction for direct mutation and that for reverse mutation, should approximately equal to 0. The other is that the correlation between them, R, should approximately be equal to -1. Now the question is whether existing methods satisfy this anti-symmetry property. I took this table from a study published in 2018 where the authors compiled a dataset called ISCB consisting of 684 mutations, half of which are direct mutations with experimentally measured delta delta Gs, and the remaining half are the corresponding reverse mutations with delta delta Gs determined according to the anti-symmetry rule. Using this dataset, they evaluated 15 predictors. It turned out that none of these methods fully satisfied the anti-symmetry property. Even in the best case, the method called pop music sim, it has a correlation far from -1. The other aspect is that in order to get a robust estimation of the performance of a method, protein level homology between the training set and the test set should be removed. This is because a method is likely to perform better on proteins that are homologous to those it was trained on, leading to overly optimistic estimation. This issue is less well recognized in the literature of delta delta G prediction. For example, here are 2 Venn diagrams showing the amount of overlap at the protein level between the ISCB dataset and 2 datasets widely used to train some of the existing methods, namely the S2648 dataset and the VariBench dataset. To address these 2 aspects, we implemented a rigorous procedure. We started out with a dataset called Q3421. The numbers here indicate the number of unique mutations in these datasets. We first removed the proteins that are homologous to any proteins in the ISCB dataset from Q3421 to get an intermediate dataset called Q1744. We then augmented Q1744 by creating a data point corresponding to the reverse mutation for each mutation in Q1744. In the end, we had a training set that is balanced for both stabilizing and destabilizing mutations with no homology to the test set. Now, let's take a look at how ThermoNet performed. We first look at whether ThermoNet can predict delta delta Gs for both direct mutations and reverse mutations. Again, this is on the ISCB test set. As can be seen from these 2 figures, ThermoNet achieved comparable performances on both direct mutations and on reverse mutations, and the performances are comparable to the state-of-the-art method pop music sim. This suggests that delta delta Gs can be predicted using 3D convolutional neural networks from raw protein structures with minimal manual feature engineering. We then look at whether predictions made by ThermoNet are antisymmetric. In this figure, we are plotting ThermoNet predictions on reverse mutations versus its predictions on direct mutations. As we can see, it achieved remarkable performance in capturing the antisymmetry of delta-delta G, with an average bias close to 0 and a Pearson correlation close to -1, substantially better than pop music scene. This suggests that ThermoNet is not overfitting to predict more stabilization or destabilization. Now, let me briefly summarize what I have just presented. We've demonstrated that delta-delta G can be predicted with 3D convolutional neural networks by treating protein structures as if they were multi-channel 3D images. We also demonstrated that ThermoNet captures the antisymmetry property of delta-delta G after being trained with the structures of both direct and reverse mutations. We also described the procedure to robustly estimate the performance of ThermoNet by homology reduction and training data augmentation. I started out working on this project in Professor Mark Gerstin's lab at Yale University and continued to work on it in my current position, so I'd like to thank Professor Mark Gerstin for his continuous support and insightful discussions, and Dr. Yuchen Yang in his lab for contributing to the early development of the project. I'd also like to thank my current advisor, Dr. Tony Capra, and the entire Capra lab at Vanderbilt University for many discussions that brought the project to the current state. And this is the end of my presentation. Thank you for watching. Please let me know if you have any questions.